Hello, everyone. Good evening to all. Thank you very much for joining us once more for this session of the BioNordika HTG molecular webinar series. This will be our third session on COVID-19 pathogenesis and findings from lymph nodes. We have today Selina Gertner and Professor Alexandar Tzankov, who will be our guest speakers. I would like to thank you both first and foremost for being with us today. I would like to say to everyone, feel free to leave your questions in the questions pane. You can write them or you can voice them afterwards. I can unmute you during our Q&A session. Also, the presentation from Professor Alexandar Tzankov is available in the handouts tab, so you can just get it, the PDF from there. Just click on it and download. Any questions or something you a bit less than perfect you experienced during the webinar, just send me a quick email or contact me via the chat function so I can try and solve it. Without further ado, I'm going to let Selina take the stage. Thank you very much. Enjoy.
Thank you all for joining this webinar. I'm really excited to co-host with Miguel this webinar here today, and excited about this topic today on COVID-19, and to have Alexandar Tzankov from Basel here as our guest speaker. Oh, let me go to the next slide. Need to go back to the pointer. Here is the agenda for the discussion today. I will start with an introduction into HTG-seq and the application. So my name is Selina Gertner. I'm Director EU Medical Affairs, Marketing and Customer Support for HTG Molecular Diagnostics. And we will then have Professor Alexandar Tzankov, Head of the Department of Histopathology and Autopsy at the Institute of Medical Genetics and Pathology from University Hospital Basel in Switzerland, speaking about the lessons learned on lymph node analysis in COVID-19 pathogenesis. We will then have a Q&A sessions, so all the questions you can already type in the chat function or the question section, and we will address them at the end. And we will then have a closing remark where I will highlight the next session of this webinar series. So for those of you already familiar with HTG, I have something new to announce, so stay tuned on what news we have. Let me introduce HTG-seq and what HTG does. So where HTG positions itself is on the RNA as a molecule. The RNA, as you know, sits right in the middle of the DNA and the protein, and DNA is a very valuable biomarker for disease analysis or the development of different diseases. But the challenge with the DNA is simply that often you don't have an actionable mutation you can go after. So while DNA is very stable and easy to analyze, the challenge is often that you do not find anything on the DNA level that gives you some insights into the biology of a disease or the disease progression and development. Protein, on the other hand, is the executor of the signal, and it's also therefore a very valuable biomarker and perceived also as a gold standard, because it really shows what's happening in the biology of the cells or in the disease. Difficulty with protein is the multiplexing, so you cannot really look at many different proteins at the same time. And RNA is combining these two aspects, so it really shows on what's happening in the cell right now, what is transcribed from the genome, and therefore it's really showing the mode of action of the disease and what is driving it. And that's why it's making it a very valuable marker for precision medicine. So what HTG provides is the HTG system. It's a device that integrates in the traditional RNA-seq workflow, and it simply automates and simplifies the library preparation. And what HTG provides is all the reagents that come with it, and there's also a laboratory service that can do the whole analysis for you. For all approaches, or for both approaches, there's also the data analysis solution available, so that it integrates nicely and you can easier analyze the gene expression data that you are generating. So let's take a closer look at the workflow and where is actually the benefit in comparison to, for example, traditional RNA-seq or some other methods. And one of the key advantages in the sample preparation step. So the challenge that we see also often when we discuss with pathologists is tissue availability. So there is a trend to less and less invasive approaches. There's less and less material available. So they like to say tissue is the issue. So you might have only a small biopsy material that you can work with, less and less resections are available, or there's even a trend to go to liquid biopsy so that you only have a blood draw and you would like to make some conclusions based on that. And HTG is supporting that trend by really using a little material. One single section of PE biopsy is sufficient, but also we can work with several different liquid biopsies. In this particular study, we were here in COVID-19, lethal COVID-19, we have to say, we're talking about autopsy material, so tissue availability is not the issue, but the integrity of the material. So if you have highly degraded material, and also in autopsy cases, it can be challenging to have intact mRNA that you want to detect. And that's where the other advantage comes into the picture I will speak about in a bit. So here's a list of sample input that we're using, and you can see for FFPE, I was mentioning one biopsy, so a section of 5 micron and a tissue area of 6 to 11 square millimeter is what we require. And you can see also here the old archival FFP blocks are listed because that's one of the key advantages the technology has, as it can work really nicely on archival FFPE material, which is highly degraded on the mRNA side. And also the different liquid biopsies, so PAX-gene blood or plasma serum for microRNAs is something we can work with, but also cells, sorted cells or cell lines. If there's RNA on stock, we can work with it, but we always prefer actually to work from the crude sample itself. So this is here an example illustrating some of the examples of the FFPE material we can work with, and in comparison you can see some of the traditional methods need either resection material or several sections to extract sufficient material, sufficient RNA to analyze them in their technology. And the other key advantage is that we work extraction-free, so there's no RNA extraction required. You simply detect your RNA biomarkers in the lysate of your sample. And why is this an advantage? You can see here the standard RNA extraction kit. For example, here for FFPE, there is an extraction kit that does purify fragments that are above 70 to 100 nucleotides. So it means if you have a highly degraded samples, which can be the case for archival FFPE tissue, you might lose many of your RNA transcripts that are simply highly degraded because they're smaller than 70 to 100 nucleotides. If you're using a standard mRNA extraction kit, it's even larger the impact because you are enriching 200 nucleotides or larger fragments. So anything higher fragmented or smaller fractions you might not be able to detect. And the quantitative nuclease protection assay, which is the chemistry that HTG uses, has the advantage that only a 50 base pair fragment needs to be present in order to detect it, so that it works really nicely also with highly degraded samples. Also, what you need to keep in mind is that the more you manipulate your samples, the more you might lose some material. So the gold standard RNA extraction kit do assume 20 to 30% loss of the RNA that you're extracting. So if you are having now your valuable sequence of interest in that material that is lost, obviously then can impact your results and can alter the conclusions that you are drawing. So in the next step, that's the HTG device that automates the library preparation. And what happens in here is the so-mentioned quantitative nuclease protection assay. And the chemistry is very robust, it's well established, and it has been around for quite some time, older than qPCR. But what we did is simply develop an automation around it. And what basically happens here in the HTG device is that we add a protection probe hybridizing to our target sequence in the lysate of the samples. And you then have here RNA-DNA hybrid. The protection probe is a DNA probe. And the S1 nuclease here digests any single-stranded molecule, so anything that did not hybridize. And in a base hydrolysis, the RNA is removed so that what comes off the HTG device is now a protection probe that did hybridize to a target sequence in the initial step. And this protection probe is then here added with sequencing barcodes and adapter. You can then quantify the samples, pool it, and load it onto your next generation sequencer. Also, the data analysis is one of the key challenges that customers might face when they do gene expression analysis, and that's why we provide an integrated data analysis solution. This tool basically allows you to do some initial QC analysis, so to see how good the quality of your data looks. You can also set some filters for the new transcriptome panel on the low expressed side of things. You can do a correlation plots and here differential expression plots, so heat maps or volcano plots or box plots, and even PCA analysis to see how your samples are clustering. So it really saves some scarce bioinformatic resources, and it provides some easy access insights into the first analysis that you can do. So just to conclude here, in a comparison to traditional RNA-seq, you can see that the workflow is much shorter. There's fewer steps, and the majority of the steps are automated, which is why the hands-on time is reduced, and also the total workflow time. So we can run an HSeq workflow in about 3 days, where traditional RNA-seq needs 8 or more days, depending on the complexity of the bioinformatic. So you not only save material, so samples that are precious, but also labors and cost on the analysis of your RNA. So I would like to briefly show you here the applications we have. You can see there's a number of panels that we provide. And basically, it simply works in a way that we designed the probes with a specific number of probes for different biological questions. That's why we have also a number of oncology-related panels, but also here an immune response panel that was mainly used in a COVID-19 study. And I would like to highlight here, in particular, those 2 panels. That one panel is looking at almost the whole transcriptome with close to 20,000 transcripts of mRNAs, and the other panel is looking almost at the whole myrinome with 2,083 human microRNAs, and therefore they are the panels that are providing a very broad overview and insight into the biology of your sample. And here are the news. What we can do now is basically to do both from one sample. So you can look at the transcriptome on your sample, and the remaining material of your lysate will then be used for the microRNA analysis, so that from one sample you get 20,000 mRNA transcripts analyzed, and the leftover material can then be used to analyze 2,083 microRNAs, and therefore you can get a very complete picture of the biology of your samples on the expression side of the mRNA and microRNA, while using the advantages that I just highlighted. And what is important to mention, our microRNA assay was stated here in Cell by Goday et al. In 2019 to be the most reproducible technology with the least detection bias compared to other technology. So therefore, it demonstrates it's a very robust chemistry, and in particular for microRNAs, a very important attribute. Now in the COVID-19 study, we will hear more details from Professor Tzankov. He will highlight in particular the findings on the immunohistochemistry side, but also on the gene expression side, and the panel that was used was here the immune response panel. And with that, I'm very pleased to introduce Professor Alexandar Tzankov from Institute for Medical Genetics and Pathology from University Hospital Basel. And he's going to present to us his molecular findings by analyzing pulmonary drained lymph nodes of COVID-19 patients. And while he's sharing his screen, I'm introducing him further, that he is the senior consultant of pathology. He is head of histopathology at Basel, and he is also very well specialized in hematopathology and a member of numerous national and international scientific societies, including the European Association of Hematopathology, European Bone Marrow Working Group, and here is the incoming chairman, as well as the International Council of Standardization in Hematology. So very pleased to have Alexandar Tzankov present here today his interesting findings on COVID-19.
Thank you very much, Selina. I hope that the technique worked and you can hear and see me, or at least see the presentation, which is more important than the image of myself. Would you
Looks
please?
Good.
Yeah, thank you so much. So, dear participants, thank you for your interest to go with me through this presentation. I would like to share with you our opinion on the morphology, the phenotype, and the gene expression profiling of the draining pulmonary lymph nodes of COVID-19. So our institution was among one of the first to systematically conduce autopsy studies on victims dying from COVID-19. Actually, in the period of March to April 2020, we were able to collect over 20 autopsy cases and publish them in highly ranked journals. And based on the morphological presentations and gene expression profiling and immunophenotype, with the help of many colleagues throughout the world, we were able to drive a kind of an idea how the pathophysiology of acute pulmonary injury in COVID-19 may look like, starting with the infection of pneumocytes type 2, but also endothelial cells by SARS coronavirus 2, deposition of the spike antigen, especially in microvasculature, then recognition of the virus, overexpression of interferon signature genes, consecutive overexpression of genes related to angiogenesis and especially thrombosis, then the microthrombosis of the pulmonary capillary network, the downregulation of important molecules responsible for the interconnections between endothelial cells, the activation of neutrophilic granulocytes within the vessels with generation of netosis, and finally the M2 polarization of macrophages and then the starting of a process called intussusceptive angiogenesis that finally disturbs the microcirculation of the alveoli. So this is how we nowadays think the pulmonary injury in COVID-19 occurs. Within this study, we also quantified the viruses in the pulmonary tissue that we obtained, and here is a log scale of the PCR analysis of those patients. And as you may see, occasionally patients were able to even expose the virus, clean the virus, in spite of dying of COVID-19. So, and there were like two groups: patients with rather high viral load and patients with a rather low viral load. And in the principal component analysis, the COVID-19 patients not only clustered apart from control cases and other pathology, lung pathology cases, but there were like two groups, so-called low viral load patients and the high viral load patients. So there were like two distinct clusters. And of course, there were some up and down regulated genes along this lung pathology. And when performing clustering analysis, finally, the biomathematical modeling told us that there are like two groups, indeed two groups, even from the perspective of gene expression profiling of COVID-19 patients, namely the high viral load patients and the low viral load patients. And one of the most important difference between those clusters was the overexpression of interferon signature genes in the high viral load patients, and the inverse correlation from a clinical pathological point of view between the interferon signature and the hospitalization duration, namely patients with high interferon signatures and high viral load at shorter hospitalization times than those with low interferon signature. In addition, as you may see here, the COVID-19 cases clustered apart from the controls, which is quite impressive. So we also linked the gene expression profile of the interferon signature genes to some genes and molecules that we were not only able to measure by means of gene expression profiling, but just also by immunohistochemistry. And indeed, it seemed that all patients with the low interferon signature had higher amounts of inflammatory or lymphoid cells within the lung tissue. They were in a more advanced stage of diffuse alveolar damage, so they had quite an immunopathology in their lungs, and more advanced, morphologically more advanced disease, more active disease, active with respect to the participation of immune cells. In addition, there was an interesting correlation between the expression of some complement compounds and those interferon signature genes, and this could also be highlighted by respective immunohistochemistry, which is quite an advantage. So looking at those things by two various methodologies for sure confirmed the robustness of the data. So based on that, we think that especially complement inhibition may be more active at later stages of diseases, and this may explain why, in spite of some clinical studies on complement inhibition in COVID-19, it seemed not to work that good, but probably the time point to expose patients to these drugs was not properly chosen. So based on those observations, in 2020, we forecasted that COVID-19 is, at least from the perspective of pathology, kind of bimodal disease. And if this is the line of incompatibility with life, there are two periods of time at which individuals are at high risk to die of COVID-19, especially fragile individuals. They are at quite a high risk to die at the initial phase of virus infection. They usually have high viral load, high interferon signature gene expression profiling. Then the viral load decreases and is even eliminated, but here the effects of the interferon signatures starts to be seen. Of course, interferons induce a lot of changes in the communication between immune cells and cells of the innate immunity. And in the second phase of the disease, like on day 12 to 14, which is quite fragile, quite a significant also clinical point, the immune infiltration and the immunopathology as well as coagulopathology seem to prevail, and once again turn at risk patients to die of COVID-19. So this is also something that has been thereafter quite consistently seen in the clinics. Well, at that time, we were also interested in what is happening in the lymph nodes of COVID-19 patients, looked for the literature, and at least in the mid of 2020, when we started composing this review paper, there was not a lot of evidence, especially no in situ evidence. There was some indirect evidence of what is happening with the immune system and the immune response in COVID-19. On the one hand, as already said, the overexpression of interferon-stimulated genes, the leukocyte traffic into the lung, which is probably to a great part responsible for the lymphopenia that is observable in severe COVID-19. Then some specific changes in the antigen presentation, but especially the activation of T cells without germinal center formation. And there was a nice paper explaining that the follicular T helper cells are quite dysfunctional in COVID-19. And of course, the plasmablasts, yet not seen, have been forecasted by means of gene expression profiling or flow cytometry phenotype to be quite significantly increased in COVID-19. And as you may later hear, there's a lot of body of evidence with respect to the effects of those plasmablasts. In addition, especially in children and in patients not suffering from severe COVID-19, some macrophage activation syndromes have been observed in COVID-19, and of course they can and may be linked to immunopathology, as you may see later on. Therefore, we decided not to only look at the lungs and lung tissues that we collected at the autopsies, but also at the lymph nodes, draining the lungs, especially of patients from the first wave of COVID-19. The major advantage of these 22 patients is that at that time they have not been treated by high-dose dexamethasone, and especially dexamethasone is of course considered to significantly alter the composition of immune cells and of course also the gene expression profiling. So except for one of these patients, none has been exposed to dexamethasone. We looked for various controls, and these controls have been chosen based on what we have observed first in the lymph nodes of COVID-19 patients. Therefore, infectious mononeucleosis and hemophagocytic lymphohistiocytosis were included, as well as draining lymph nodes of diffuse alveolar damages caused by other causative agents. Of course, all these data have been performed like in duplicates. On the one hand, immunophenotypization in situ with means of immunohistochemistry, and then of course the gene expression profiling. And therefore we feel that the data generated is quite robust. There was some unavoidable differences between the controls and the COVID-19 patients. Of course, the median age of the patients, and also the exposition towards immunosuppressive therapy. Since we have been looking for cases of EBV infection in adults, of course, they were slightly overrepresented in the control collective. And as I said, only one patient here has been exposed to high-dose dexamethasone, and for obvious reasons, because lymphopenia is a kind of a symptom of this COVID-19, especially of severe COVID-19. Consider the median lymphocyte count in COVID-19 was lower. Let's start with the morphology. We were quite astonished to see that the lymph nodes look quite different from lymph nodes that we used to observe in reactive or virus-induced processes. Instead of a parafollicular expansion, we see here a considerable hemostasis, edema, dilated sinuses, and actually no germinal centers. So there's just one, and here one more, there are two germinal centers in these lymph nodes. So this is quite impressive, given the fact that we're expecting that our immune system has to start to produce antibodies, highly selective antibodies against COVID-19. So this is a different kind of a presentation. We're looking here at COVID-19 cases and controls, comparing the presence of extensive edema, stasis and edema. This is quite significantly more common in COVID-19. Increased amount of plasmablasts, also quite significant in COVID-19, or very few germinal centers, if any, also quite significant, more common in COVID-19. Here, also the background cells of the germinal centers, the folliculodendritic cells, as yielded by immunostaining for CD21, were quite decreased and disrupted in COVID-19 compared to our control collectives. One very consistent, I would say one of the most consistent findings in all these COVID-19 lymph nodes was the high amounts of plasmablasts, these ugly-looking cells, and this is a special way of an alternative B-cell stimulation, on which I'm going to talk in a second. These plasmablasts either express IgM or IgG, as demonstrated here. Another consistent finding was the high occurrence of histiocytes with increased hemophagocytic activity. Those cells here looking like strawberry cells with representing histiocytes with engulfed erythroid cells. So this is quite a significant hemophagocytic activity. And indeed, one of our patients of this collective also displayed a full-blown hemophagocytic lymphohistiocytosis that complicated his COVID-19 disease course. So to summarize, edema, a lot of plasmablasts, and these plasmablasts have been studied many years ago by the group of Würzburg, demonstrating that these plasmablasts are parafollicular activating of B-blast reactions is quite common in mucosal lymph nodes and represents a rapid immune response with generation of high levels of IgM, occasionally IgG, that are not very highly selective and usually active against or microorganisms containing capsules. So quite an unspecific but very rapid immune response that does not generate immunologic memory. So this is something that we observed also in our lymph nodes. We of course also quantified the viruses, SARS coronavirus 2 in our lymph nodes, and detected that in 14 of 20 cases there were detectable genomes, and the detectable genomes suggested a virus concentration about 100 times lower than in the lungs. So obviously this is not this site of infection or of a generation of viruses, but the battlefield, a battlefield of the immune system, as you may see later. There were also 6 negative cases, and this negativity correlated with the duration of hospitalization. The longer the patient had been hospitalized before dying, the higher the chance of having eliminated the virus, and they correlated also with improving differential white blood cell counts. What we were quite astonished to see is that actually, except of one case, we weren't able to identify SARS coronavirus 2 antigens in these lymph nodes. This is here antharcotic pigment, typical for city dwellers in Europe. But here the red staining yields the N antigen of SARS coronavirus 2, which is probably brought here to the lymph node by histiocytes, since this case did not display a very high very low CT values considering virus amounts. What we were astonishing to see is that in 4 patients there were significant amounts of EBV-infected lymphocytes, and also in 2 cases significant amounts of herpes simplex virus-infected cells, suggesting an immunosuppression. Considering the different T-cell compounds, there was a decrease of T helper 1 T-beta equivalents in the COVID-19 lymph nodes, a significant decrease of FOXP3 regulatory T-cell equivalents in the COVID-19 lymph nodes. So this is the COVID-19, this is the amount in the control cases, and quite a significant M2 polarization of macrophages, as one may estimate from comparing the stainings for lysozyme, which is rather M1 marker. This is the normal lymph node, the COVID lymph node, and CD163, which is an M2 marker. This is a COVID lymph node, this is a normal lymph node, and finally resulting in a quite decreased M1 M2 ratio in COVID-19. In addition, there was highly neoangiogenic activity in the COVID-19 lymph nodes, as yield here by estimation of the amount of CD105 positive newly formed vessels. This is a staining that we made after we got the results from the HTG, which I'm going to share with you in a second. Also, a considerable amount of von Willebrand factor positive mononuclear cells in the lymph node compared to the normal lymph nodes, where the von Willebrand factor is physiologically only found in the endothelial cells. So let me share with you the gene expression profiling data, starting with principal component analysis. So as you may see here, the control cases in red and COVID-19 cases in blue, they clustered apart, suggesting that the response we're seeing in these lymph nodes of COVID-19 is something rather genuine. There were only 2 control cases that clustered in the vicinity of COVID-19. The one is hemophagocytic lymphohistiocytosis, and one draining lymph node of a complicated pneumococcus pneumonia. Let us look at the top 20 up and down regulated genes in COVID-19 compared to control. So COVID is always blue. And as you may easily see, among the 20 upregulated genes, there is not a single gene related to immune stimulation or T-cell function. Indeed, genes related to platelet vascular functions, angiogenesis, and thrombosis were amongst us the most upregulated genes in COVID-19 compared to controls. And here, the von Willebrand factor appears. So the gene with the most significant p-value of upregulation was actually von Willebrand factor. This is why we stained the lymph node for von Willebrand factor and could correlate these findings with these observations here. Among the downregulated genes, one may see a lot of genes related to T-cell functions, and this is quite compatible to what we have seen by means of immunohistochemistry with the various deregulation or disappearance of distinct T-cell populations. In addition, antithrombotic gene plextrin was also downregulated in COVID, which makes sense, of course. And an important couple of genes, ICAM3 and the integrin beta-2 gene, which are the receptor and the ligand, were also downregulated in COVID-19 compared to controls, which is something that I'm going to talk on in a second. We performed also pathway enrichment analysis, comparing COVID-19 to controls, and linked to the top-down and upregulated genes. We have seen that B-cell receptor signaling, graft versus host disease, or lysozyme function, which is roughly the function of M1 polarized macrophages, were all decreased in COVID-19, while early response genes, genes related to nuclear division, chromatid segregation, and all the mitotic cycles were quite upregulated in COVID-19. In addition, we used the predefined gene sets in order to look whether COVID-19 cases cluster apart from controls when we only concern genes related to angiogenesis. And indeed, as you may easily see here, the COVID-19 cases rather perfectly cluster apart from the controls based on the expression of these 20 genes related to angiogenesis and to a part of thrombosis. So there's a clear angiogenic signature in COVID-19, which is quite compatible to what we know now from the lungs, that severe COVID-19 is angiocentric syndrome with high propensity towards coagulation. We also performed some analysis enriching customly genes. So actually, all these genes that you see here have been also stained by immunohistochemistry. We use these markers in order to see whether what we see in immunohistochemistry can be reproduced by gene expression profiling and vice versa. And these are genes, for example, related to plasmablast functions like IRF4 encoding for MUM1 or MZV1, or here XBP1. Here the gene for CD20, here the gene for CD21, the follicular dendritic cell marker that I have shown you. And here other genes related to various B and T cell functions, like for example TBET here, TBX21. And based on that, there was also a nearly perfect clustering of COVID-19 cases apart from the controls, increasing the robustness of the data that we have shown to you previously. We also used our biostatistical approach, professional biostatistical approach, apart from the software provided by HTG, in order to test whether all what we see holds true. And indeed, in this unsupervised clustering analysis, the COVID-19 and control cases really clustered apart, except for these two. And actually, these are the two cases that I have shown to you in the principal component analysis. And if you look at the various genes that are under and overexpressed in this clustering analysis, then you may figure out genes related to T cell functions, genes related to M2 polarization, to platelet and vascular functions, to arachidonic acid metabolism, which are under or overexpressed in the various groups, and show a rather consistent signature pointing towards this important processes that seem to be significantly deregulated in COVID-19. Probably I would like to drive your attention to this gene, prostaglandin synthase, which I'm going to discuss in a little bit more detail, and probably also to the PDGF receptor alpha that seems to be quite overexpressed in COVID-19 compared to controls. So this prostaglandin D2 synthase is an important enzyme that converts prostaglandin H2 to prostaglandin D2. And the prostaglandin D2 has very important functions towards platelet inhibition and bronchodilation. And this more or less the counterpart of prostaglandin I2 and prostaglandin E in that consideration. There are interesting models, knockout models, showing that a knockout of this gene leads to severe lung damage, neutrophilic influx, and increased vascular permeability, basically processes that we observe in the lungs and roughly observed in the lymph nodes, except, for example, the edema and the capillarostasis that has for sure something to do with the vascular permeability. It is interesting to know that the prostaglandin synthase, D2 synthase, works quite optimal if the level of PGH2 are low. So if the PGH2 is generated by COX-1, then this prostaglandin synthase works optimally and can convert the PGH2 to prostaglandin D2, while if the enzyme is confronted to very high amounts of prostaglandin H2 generated by COX-2, then the preferential way of conversion goes towards PGA and PGI2. This has been shown many years ago in a completely different conditions in some asthma studies. And interestingly enough, from the very beginning of the pandemics, there were interesting reports showing that the COX-2 inhibitor treatments may be promising in COVID-19. There are several studies that include a little bit more patients, but the best controlled study is the one here from Singapore. Unfortunately, for several reasons, this drug is still not considered standard of care. But I think some data are interesting and might prompt us think of properly designed studies. Another gene that was quite overexpressed in this COVID-19 lymph nodes was the platelet-derived growth factor receptor alpha. And you may know there are a lot of inhibitors coming from oncology, which may certainly be thought of being implicated also here, especially the non-mutational selective ones. Well, let's summarize what were we able to learn from these lymph nodes of COVID-19. So obviously the immune response is quite pathologic. There is evidence towards skewing towards plasmablast formation, and these plasmablasts are known to produce suboptimal antibodies, especially respecting viruses. Such antibodies may, on the one hand, foster antibody-mediated enhancement and, overstress or exhaust macrophages, but they also may foster virus uptake in monocytes and macrophages, which is thought to be one of the mechanisms of viral spread throughout the body. There's an obvious failure to mount germinal center reactions and select high-affinity antibody-producing plasma cells in patients that suffered from lethal COVID-19. So, as I used to say, I would doubt whether, if these patients wouldn't have died of COVID-19, would be able to mount a very efficient, at least antibody-dependent immune response towards COVID-19. There is evidence of severe T-cell deregulation and suppression. An indirect evidence is the reactivation of other viruses of the herpes virus family. But considering the deregulation and of transcriptional factors like TBET and FOXP3 and the decreased amount of respective cells like T helper 1 cells and T regulatory T cells, may pave the way towards autoimmunity. In addition, just consider that neuropilin-1, that is an alternative receptor for SARS coronavirus 2, is also expressed on Tregs, and we still do not have enough data in that respect. There is evidence for macrophage activation, for pathologic macrophage activation and M2 polarization. This has been shown by many studies, by considerably better studies than ours, and to a part, of course, explains the activation, macrophage activation syndromes observable in COVID-19, and also paves the way towards a pathological tissue repair. There are unexpected gene expression profiles that nicely explain what we see here, but also extend our knowledge and drive our attention towards interesting targets. Overexpression of genes related to platelet and vascular functions and angiogenesis. One would not really expect that in a virus disease. Downregulation of genes related to T and NK cell functions, and some genes related to leukocyte adhesion and migration. So by means of this, the most important findings in the draining pulmonary lymph nodes in COVID-19 is the extrafollicular plasmablast activation with production of not very functional antibodies suggested, the obvious T-cell dysfunction, multiple level of evidence, the M2 polarization, considerable body of evidence from our and many other studies, some hints towards a reduced hemotaxis of leukocytes, and very unexpected gene expression profile with high expression of angiogenesis genes and genes encoding for factors related to hypercoagulability. By means of this, I would like to thank you for your attention and thank you for giving me the opportunity to share our data, and I'm ready to take your questions.
Thank you so much, Alex, for this great presentation and for providing a beautiful overview of your findings on COVID-19. So really interesting how you also illustrated how the gene expression profiling educated or confirmed in the first way what you found on the immunohistochemistry on the protein level, but then also, for example, with the van Willebrand factor, it educated it further, so you revalidated and confirmed it on protein level. So really interesting and beautiful how you went about discovering that. So
there is one question, but for whatever reason it's so small in my I cannot really see it.
Yeah, so I can, or Miguel, you want to go ahead?
Richelli.
I can read the questions out for you then, if it's okay.
Yeah.
The first question is about your findings on the interferon stimulating genes in the COVID cluster for the first slides. What was the cutoff used to assign high and low viral load?
So this was 10 to the third. So at the end of the day, we used the median of this group, but there's not a real cutoff that can be considered universal. So we just measured the amount of viruses by RT-qPCR, and then considered the median value as the cutoff, and cases over the median value were considered high viral load, and those under the median value low viral load. So quite a Solomonic solution. But it was interesting then to see the links between the viral load, the interferon response gene signature, and the hospitalization time, and finally the link between this on the one hand, and then the amount of inflammatory cells and complement compounds in the tissues.
Okay. Thank you for that. I believe the next question is a follow-up question. Could you clarify again how the definition of severe was applied here? If it was based on viral load, or if it was clinical, and if it was clinical, which factors?
Well, me as a pathologist, I only have the patients who died on COVID-19. So this is the most severe COVID-19. If you die on COVID, then it's severe. This was the definition. So we had not much access to tissues or secretions of patients considered to suffer from severe clinical COVID, but not lethal clinical COVID. So we had some bronchial alveolar lavages where we are now studying also the gene expression profiling, actually by the HTG platform. So we didn't expect that this will function, but it functioned. But we only have 5 cases, and it's a statistical nightmare to get the idea what the gene expression profiling of these few cases tells us. So there is also a question. Now I can see the questions. Since all the patients were not vaccinated, so they was for the first wave 2020, and therefore we had no vaccinated patients. Now we have occasionally patients with vaccination who die on COVID-19. Usually, so in the third wave we had a lot of unvaccinated patients who died, younger unvaccinated patients who died, but unfortunately only a few have given consent for autopsies. And now in the 6th wave, more or less, we have occasionally vaccinated people dying of COVID-19. These are usually very fragile elderly patients, and until now we have experience with 2. Both of them died of infectious complications. There was some interesting observations in the 2nd wave when dexamethasone started to be broadly used, and there we had some autopsy experience, and there the infectious complications, especially fungal pneumonias, were more common than in the 1st wave. So the effects of dexamethasone, they cost something, so it's not for free as anything in life. Yeah.
And so in regards to that question, what do you expect your findings to look like in a vaccinated person? Would that have a different impact?
We haven't systematically looked at the lymph nodes, so at least the two vaccinated persons had optically more or less the same changes as the unvaccinated from the 1st wave. But as I said, the 2 were very fragile, over 85, with a lot of comorbidities, and especially if you think of COVID-19 as a B-model disease. At the first possible hit is driven very strongly driven by the virus, and if the vaccination has not been able to induce a very efficient protective immunity, then these patients just die along the viremia or along the pulmonemia of viruses. So this may be the case.
Okay, thank you.
Thank you.
There's another question also by Peter Riccelli. Is it clear at this point whether the unexpected dysregulated pathways are altered due to the host response in the pathways you expected, or is it driven by the viral genome machinery? So I'm not sure that the viral genome machinery plays a very important role. Probably, maybe the spike antigen plays a certain role, but for sure the target cells. So at least in our model and the way we're thinking of is that in severe lethal COVID-19, the main target cells of SARS coronavirus are the pneumocytes type 2 and the endothelial cells. And then the virus proliferates in these cells and stops their physiological function. And the readout of this is, of course, the liberation of von Willebrand factor, of angiopoietin 2, and so on and so on. And then the body tries to mount a certain immune response against that. And the macrophages obviously play a very important role. So beyond this very root M1, M2 polarization, there are various types of macrophages, including macrophages that try to substitute endothelial cells. So they really try to express some markers of endothelial function. And these macrophages, of course, produce sets of cytokines and biologic actively substances that finally lead to severe immunopathology as well. So it's not completely clear, but there's a central role of macrophage degeneration in orchestrating all these things that we see here.
Okay, thank you. I think we went through all the questions. If there's any more questions, please ask them now. So thank you again for this great presentation, Alex, and for the great discussion, really valuable findings, and helps us understand COVID-19 better. And was really nice having you here. Thank you so much.
Thank you.
Thank you
Goodbye.
for taking the time.
Goodbye.
Thank you.
So I will just use the opportunity to announce the next webinar. And the next webinar will be coming up on April 26th. So we will have Paunima Patil from Institute of Human Genetics from University Hospital Ulm in Germany presenting about her findings on oncogenic microRNAs that are deregulated in T-cell polylymphocytic leukemia. So we will have a leukemia topic and microRNA discoveries here. Save the date for the time being. We don't have the link ready for you to register yet, but you will be receiving an information about how to register and connect to this event. So thank you again for joining us today. Thank you, Miguel, for co-hosting. Well done as always, and happy to see you here next time.
Thank you very much, everyone. Any feedbacks, you can send them through me. I will be releasing the recording of this session soon enough. Okay. See you on the 26th, if not before. Take care.
Thank you.
Thank you for today. Bye.